Alrighty, welcome to me finally having access again. So let me go ahead and start this just by saying we're going to finish up chapter 8 here and jump into chapter 9 a little bit. I know this is asynchronous, so it might be a little odd. I'll try and go nice and slow and I'll break it up. I don't think this will be a full hour and 15-minute lecture. But I just did want to finish up chapter 8 and get into some of the applications and techniques in chapter 9 for nucleic acid technologies. So I'll start by pulling up where we left off in chapter 8. We had talked about the various techniques, denaturization, melting point, some of the applications for antisense therapy. But chapter 8 kind of finishes up talking about different ways that we can undergo mutagenesis to our DNA or RNA. So I want to talk about just a few of these fairly briefly. There's not a lot here, but one reaction that we typically see is deamination. This is relatively slow, and essentially we lose an amine group and replace it with an oxygen. Okay. The thing is, we have a really large number of residues that have amine groups in here. So even though this is a slow reaction, in effect, we end up converting about 100 cytosines to uracils every day in a cell. That's in each cell. Okay. We can also see something like methylcytosine getting converted to thymine, or adenine going to hyperxanthine. More of that in metabolism later on down the road, like next semester or whenever we have Biochem 2 again. But the idea is we see this conversion taking place. This deamination results in a mutation. So you can see if you have an RNA, say like an mRNA code, and it should be a C, it gets converted to a U. So that can introduce a protein mutation as well. Right, so this occurs. We'll talk about what happens when it occurs in a little bit. We can also see depurination as well too. This is a pretty significant event that happens here. That N-glycosidic bond that connects the purine to the ribose gets hydrolyzed, and it releases the purine. Okay, so in this case we have guanine here. It gets cleaved off of the backbone sugar, and we have this apurinic residue, which basically means if you would normally have a GC interaction, suddenly you're missing your G, so there's nothing to hydrogen bond there too. This is pretty significant. We can see about 10,000 purines that become hydrolyzed each day in a cell. Okay. We can also see that these mutagenesis can occur through chemical means as well too. We can have some oxidative damage that results in this. We'll often see in this case a hydroxylation of guanine. So we have basically this oxygenation reaction. We're adding an oxygen to it. Mitochondrial DNA is the most susceptible to this. Okay. We can also have alkylation. We'll see methylation of guanines in particular in there as well too. But the important thing to note is with all these mutations, there are repair mechanisms that will correct most of them. A lot of times, as long as you do not see a significant number of mutations, you won't see any significant effects in there too. If you look at something like exposure to high levels of radiation, you end up overwhelming the repair system, and that's where you end up with radiation sickness and long-term damage and diseases in there. Or sometimes you will, if you have like probably about double-stranded DNA, you might not have a complete parent strand, unbroken parent strand or unmutated parent strand to compare and fix. So we can see damage there too. The accumulation of oxidative damage is something that might result in aging. Okay? If we look at a few alkylating agents in here, we can see like a dimethyl nitrosamine, dimethyl sulfate, and nitrogen mustard. Maybe our mustard gas. I'm not sure if that's mustard gas or not, but I don't think it is actually. But this nitrogen mustard, I'm not going to put that on my sandwich. But it can alkylate and add our alkyl groups to our compounds in here. Okay, you can see here we have our alkyl methionine, adenylyl methionine, sorry. Yeah. All right, UV light can cause pyrimidines to dimerize, and we'll see what we mean about this in the picture on the next slide coming up. In this, UV light, if you look at people spend a lot of time out in the sun, their skin can get kind of weathered or a lot of wrinkles and damage will show up, and this may be due to a threshold exposure of UV light, and of course this could be also the mechanism for skin cancer to arise as well too. If we look at ionizing radiation, or X-rays and our gamma rays, they will cause ring opening and strand breaking, and that's really hard to fix, because if you completely cleave a strand of DNA, then you've got some problems trying to hook it back together. That ligating it back together is a specialized process. So our cells can repair many of these modifications, but some are going to cause mutations, and mutation is a fairly natural event here. If we accumulate lots of mutations, we can call that aging. You can see my nice little wrinkles here and whatever. And carcinogenesis, hopefully you can't see any carcinogenesis happening in Dr. Ebert. If you do, let me know. We accumulate mutations, and sometimes these mutations can be beneficial, but most of the time we have, a reasonably optimized organism in human beings for survival and proliferation. So when we start accumulating mutations within an individual, then we see the aging process and cancer. If you live long enough, you're going to develop cancer. Okay. Here we can see a dimerization of our purines. So if I go back up here, we're talking about this dimerization of pyrimidines. We essentially have a covalent bond that forms between adjacent pyrimidines, which introduces a kink in the DNA. So instead of twisting around like it should, we get this little flex or kink in there, and we can't make our base pairs as easily with our complementary partners. Okay. But again, as long as you have low levels of mutations, it's not too bad. We have a threshold effect, so we kind of go along and accumulate mutations, and you get to a point where all of a sudden it takes off. I joke about getting old. I'm probably much older than I look, but when I turned 35, I'm like, oh, I feel great. I turned 38, I'm like, oh, I still feel pretty good. I turned 40, I'm like, oh yeah, I still feel pretty good, but every now and then I'll get a twinge in my back, and it's just like it drops off a cliff. You can see this in professional sports where they talk about running backs in football. If you have like a 32, 33-year-old running back, they're an old man, because usually they drop off the cliff pretty fast once they hit their 30s. You see like some unique quarterbacks like Tom Brady and Drew Brees, who are both in their 40s, still playing at high levels, but most quarterbacks will end their career sometime in their 30s. I'm trying to think of a quarterback who spent 1 year too many in there, and you just see like their skill level kind of drop off. So we can see this threshold accumulation of mutations, and all of a sudden you get old. Somewhere when I was went to my doctor, I was like, hey, I have these pains and aches, and he's like, oh, I'm just getting old. It's just taking longer for things to heal due to these accumulations of mutations. All right. Our nucleotides also play other roles. We sort of mentioned this in one of the previous slides somewhere like a week ago or something, in the far distant past, called the Jurassic period or something, because it was such a long time ago, it feels like. But many of them will be used as sources of energy, and we can think of this as our ATP, our currency of the cell. Our ATP, when we hydrolyze this triphosphate bond, we get lots of energy. We can also use GTP and CTP and TTP and UTP as sources of energy. Those are less common. GTP is fairly common, but ATP is like the big one. Our ATP levels are going to be an indicator of the energy capability of a cell for the most part. And of course, we can look at our RNA sources of energy and our DNA sources of energy here, or I guess our ribonucleic and deoxyribonucleic triphosphates. Okay. So hydrolyzing, and usually it's going to be the alpha-beta bond here. It's going to give us a ton of energy, so we can couple that hydrolysis with many other processes, and that's what the body does. If we use ATP, cleave that to make either AMP or ADP sometimes to get energy to drive something forward. We saw this most recently when we looked at the actin-myosin cycle. We had our myosin walking along the actin filaments there, and that power stroke involved cleaving ATP to ADP and releasing the phosphate and releasing the ADP and binding new ATP and all that good stuff. All right, so we can get lots of energy from these high energy compounds. Our nucleic acids are high energy compounds when they are phosphorylated. We can also see nucleic acids play roles as coenzymes. Coenzyme A, it's a pretty obvious name there, right? But if we look at our base, we have this, adenosine diphosphate as kind of our base, and then we add a pantothenic acid and a beta-mercaptoethylamine to the end to get this coenzyme A. Okay, we can see NAD and NADPH are both cofactors, coenzymes that are critically important in the electron transport chain, pentose phosphate pathway. And we'll also see FAD in there as well too. I really like this one because, maybe too much Simpsons or Futurama. We got flavin nucleotides here. So, fun with those. All right. We can also utilize our nucleic acids as regulatory molecules as well too. And perhaps one of the most common one is cyclic AMP. So here we have adenosine monophosphate. Okay, and we make a 5' to 3' cyclic connection with that single phosphate that's on there, and this cyclic AMP can bind to a variety of receptors. We'll often see this as a second messenger. Was it our GTP? Is it GPCR signaling molecules? And they're they'll bind and they'll induce the formation of cyclic AMP, which will travel as a second messenger to amplify the signal for whatever these protein receptors are. We also have cyclic GMP as well too. Obviously, we could make cyclic CMP and cyclic TMP and cyclic UMP as well, but they don't seem to play as much of a role. But cyclic AMP is the big one, followed by GMP. So A and G seem to be our major players for signaling and energy. We also end up with something weird here. I just like the abbreviation. I keep those in here, PP, GPP. If you can't remember the abbreviation for a compound, just throw in a lot of G's and P's because it's phosphates and you get guanine or glucose or glycogen or whatever. Got a decent chance of having it in there. But here we can see that we have phosphorylated not only the 5' hydroxyl group, but the 3' hydroxyl group as well too. So I forget what this signals, what function this does off the top of my head, but it's just another signaling molecule, another regulatory molecule. If we go back to our enzymes, these could be allosteric effectors that promote or inhibit the enzyme's function. Okay. All right, and that's it for chapter 8. See, it took a whole week to get here, but it's a very, very short chapter. The main thing is just, understanding the function and structures of our nucleotides, our nucleic acids. Know the difference between a nucleic acid, a nucleoside, and a nucleotide. Just depends on whether you have, the base by itself, the nucleobase attached to the ribose, or the nucleobase attached to the ribose that is phosphorylated. Okay. So names and structures, I'm already working on exam 4. Promise I'll get exam. I would have had exam 3 graded last night if it weren't for this power outage and lack inability to connect to the internet. So anyway, we'll also talk about the structural basis of DNA function as well too. So again, we have our double-stranded DNA, our single-stranded RNA, and how they fold a little bit. So that'll probably be a few. Understanding how they interact with each other and the formation of hydrogen bonds that stabilize the overall conformation of whatever particular structure we're looking at is very important. We also talk about denaturization. We have our double-stranded DNA. We can kind of unzip the DNA to allow it to either replicate or to express certain sites that can be converted into mRNA, which can then be expressed as proteins in there too. And you might just a little bit about mutagenesis. Again, our mutation is just constantly a natural process. That deamination occurs, oxidative damage. We breathe oxygen. We have lots of oxygen there. That's why antioxidants are a big important thing. Clearly, I need more antioxidants for my face or whatever. Anyway, that's chapter 8. Okay. So what I want to do now, all right, is share chapter 9. Okay, yes. All right. And talk about some of the various technologies that we use with this. There are a couple big ones that are very commonly used. We're going to talk about cloning techniques. We're going to talk about expression of recombinant proteins. our products, expression proteins, maybe like our flu vaccine, which I need to do very soon. I gotta run over to Wake and do that. We'll talk a little bit about some analysis methods of DNA, and we can talk about DNA microarray technology, which I always kind of shudder because, you know, it's not that bad, but I just had a bad experience in my introduction to chip arrays. I was like, oh my Lord. Yeah, so I have a bias against them, but they're actually really useful, even if the process of making them can kind of sometimes get complex. But anyway, let's start with the idea of recombinant DNA, okay? So our recombinant DNA is just artificially created DNA. We can either mimic sequences that we have seen in nature, or we can make completely new ones, okay? But our recombinant DNA ends up being the basis of a lot of our modern molecular biology. Sorry, I'm trying to need a more comfortable chair. All right. Oh, that's a really nice room. I should lecture in this room more often. But anyway, our recombinant DNA is just something that we make artificially. Now, you know, artificially, that's, you know, kind of one of those, you know, how do we weigh that? But it's just, we're just saying that it's not something that's found in nature, although we might harness natural processes to help us make recombinant DNA. Our recombinant DNA is the basis of just a ton of stuff that happens, whether it's cloning genes, overexpressing proteins. We can look at transgenic food, transgenic animals. I think I've got a link to either Dolly the sheep. I know there's a link there to the GFP bunny and the GFP cat. They are, you know, basically animals that have been transfected to express certain proteins. Sometimes that can be very helpful. We use like a green fluorescent protein tag on a particular protein. We can, you know, track where that protein is. So for GFP bunny, he's a bunny that glows in the dark. Well, under the proper light condition. Excuse me. All right. So let's jump ahead though and talk about cloning a little bit. And if you grew up on a steady diet of science fiction, like say maybe some nerd or something, you know, cloning was a big thing where we're gonna, you know, make copies of people, you know, have an organismal cloning. Okay, so we make an identical copy of a whole organism, right? So like Dolly the sheep is a clone of a sheep that was made in, I believe, the 90s, might have been the early 2000s, but maybe in the late 90s. The organisms aren't necessarily identical because, you know, they may have different expression patterns that happen during their maturation. But going back to science fiction, there was some, you know, movie or TV show where some scientist was being chased by bad guys. So he made 12 clones of himself. So he had 12 identical hymns, whatever. And then he buys like 12 plane tickets to different cities around the world and sends the clones there. And you know, meanwhile, he stays in the city where the bad guys are looking for him, but, you know, they have access to this. Well, I always thought it was a horrible thing because no matter where the bad guys went to find him on one of these plane trips, they'd find him because he sent a clone to everywhere, you know, and so they're obviously going to kill one of his clones, which was, you know, the same as him. I don't know. It's just, I mean, that's a question for bioethics, isn't it? But anyway, the whole point is, organism cloning is, you know, our fancy sci-fi, you know, oh, it's so cool to have a clone. I'll clone myself to get all my work done. But it really is a separate organism, and it's not something that we see too often. Okay. On the other hand, DNA cloning is something that happens a lot. Okay. We may clone just a particular gene or a portion of a gene, or some piece of DNA or RNA in some cases, to, you know, fulfill some process, to fulfill some need. Okay, if we were trying to make a vaccine, we might want pieces of a particular DNA to say, hey, if you see this DNA or this RNA or this protein or something in there, you know, that's bad, you know, so attack that. Or we may want to overexpress something that can be used as medication. Okay, so for DNA cloning, we isolate usually a specific gene from our source organism, and we amplify it in the target organism. We'll see this when we'll use like bacteria to grow up and overexpress particular genes. Okay? So the basic steps for the cloning is to cut out the source DNA at its boundaries of the gene. Okay, we find a particular carrier for that. This is going to be known as our vector. So something we can insert DNA into, and then we insert the gene into the vector, and then get the recombinant vector into the host cell. All right? And we let the host cell produce multiple copies of this recombinant DNA. In my graduate work, I did something like this with yeast. We had a particular strain of yeast where we had knocked out the iron sulfur protein, and we, so that we could insert and basically infect our well, infect is probably a bad term because I'm not giving it a virus or bacteria, but we could insert a particular plasmid that contained a mutated version of the iron sulfur protein gene, and then we would let the yeast cell, utilize that DNA to produce, a mutated iron sulfur protein, and that's how we tested several different mutations to figure out, what effect these mutations had on the iron sulfur protein and the function of the electron transport chain. All right, well, let's talk about how we do this, okay? So step one, we need to make our recombinant vector, all right? So we're typically going to have some type of cloning vector. In this case, we're going to talk about plasmids. We also have cosmids and some other systems that we'll mention here a little bit. But we also have our gene of interest. So maybe this comes from a eukaryotic chromosome. So what we're going to do is we're going to use restriction endonucleases. And I always really liked restriction endonucleases. I believe I have a link to the New England Biolabs, the NEB restriction endonuclease mapping site. But basically a restriction endonuclease recognizes a sequence of DNA. Typically it's 6 base pairs long, but it can be 4 or 8. And the most common ones, they're all in a row, but you might recognize 4 here and then have a space and 4 down here, something like that. But whenever they recognize this pattern of DNA, they make a cut at a specific place, okay? And this cut can either be a blunt end where it cuts straight across, like we're slicing through a sandwich, or it can be something that's more like let's see if I can do this here, a sticky end where you have an overlap in there as well too, right? But the key is our restriction endonucleases recognize a particular site. So we want to make sure we have this particular sequence in our cloning vector, and then that we can find this sequence on either side of our gene. Sometimes we need 2 sequences in here, so we might use like a BAM1 and a ECO-R1 on either side. So we basically have 2 different restriction nucleases that we use to cut it out, and we just make sure we have those sites in here so that we can ligate in that section out. So here we have our fragments. We have cut out our gene. We see here we have sticky ends, our overhangs. So when we make this cut in here, we can insert this in and ligate it together. We'll use our DNA ligase, and we now have our recombinant vector that has the addition of this gene in there. Okay. We might have other stuff in there, so a lot of times we need to test and make sure that our recombinant vector has what we want it to have and only what we want it to have. But that's troubleshooting in there. Okay. So yeah, here's our restriction endonucleases. They cleave at specific sequences in here. Should have looked further ahead in my slides for this today. Staggered cuts are our sticky ends, straight cuts, blunt ends in there. Okay. And then here's the link to that New England Biolabs that has a list of, I can't remember when I first started doing this, we used to do restriction enzyme mapping with DNA, and you just, treat your DNA with different restriction enzymes, and you get different sized fragments that you run out on gel, and then you could figure out how to put all these fragments back together to kind of get a general order of sequencing in there. This is no longer done because now we have such advanced cloning techniques that we can just sequence the whole thing, and we don't have to try and figure this out. So now we use our restriction endonucleases in a new way. I also remember this not because I had fun doing it, because it was like putting together a puzzle in reverse, something like that. But that was where I made my first, one of my few only costly errors. I did not change tips on my pipetter, and I contaminated a $300 or $400 vial. These are tiny, tiny, tiny little vials that are $300 or $400, and this was back in, maybe the early 90s. Contaminated vial of this very expensive restriction endonuclease, and I was like, oh, yeah, and I felt bad. So that was probably my most expensive mistake as an undergrad. All right. Anyway, so if we look at our restriction endonucleases, maybe here we have EGO-R1 that recognizes this particular sequence, and it cleaves here. Okay, so you get these two sticky ends. Here's a different one, PVU2, okay, and it recognizes this sequence and cleaves to give you a blunt end, okay. So if we use those two, we want to make sure we only have one of each site in our vector, so that we get our two ends here, and then maybe our gene of interest. We see those mirroring our particular gene shown here in blue, and so we now have this these extra little ends on either side, but we have our gene, and we can ligate it into our vector. Okay, so what we've done, we've opened up our vector, inserted some additional DNA, ligated it back together, and now we have this happy little plasmid that has our gene of interest in it. Okay, so I mentioned with mutations, when you break a strand, it's really hard to repair it. Well, DNA ligase will covalently join those DNA fragments in here. So normally we see this in DNA repair. Okay, so we have a human DNA ligase that utilizes ATP as its energy source because it costs energy to do this. But bacterial DNA ligase will use NADH as its energy source. So depending on whether we are using a human-derived plasmid or a bacterial-derived plasmid, or what particular DNA ligase we're using, we'll need to know. Well, that will inform us as to the particulars that we need to do to ligate it back together. Okay. All right. So once we have our recombinant vector with our stuff in here, yay, everything's good. Okay. Now there is something to watch out for, and we'll talk about this when we get to testing here in a little bit. If I have, a plasmid that only has one cut end here, okay, and I ligate it back together, if I cut it, it might just ligate straight back together. Okay, so just having a single site, sometimes you may get your DNA inserted in there and ligated in, and sometimes you just ligate back your original plasmid. If you have two cut sites in here, I can't ligate this back together because they don't match up. I've got an overhang here and I've got a straight end here. So I need to have another piece in here that will allow that to happen. Now, of course, I cut out a piece in there, okay? So, maybe I could get my original and put it back together that way, but hopefully I'll have a much higher concentration of my DNA of interest to get that in there. All right. Anyway, let's assume we've successfully made our recombinant vector. We'll talk about ways to test that in a little bit. We need to get that into the host cell. Now, there's different ways you can do this. You can do a kind of a chemical shock, or electroporation is another way, where you basically pass a current or use a certain concentration of chemicals to induce an opening in the cell wall or the cell membrane, okay? And that opening will allow the plasmids to fit in, and then presumably it happen here. I had a friend who used electroporation a lot in her research, and so you basically have this little current that zaps through there. You got to make sure your current is just right so that you shock it and it opens up and then comes back. If your current is too high, everything explodes. So I'd often hear, she was in the lab next to mine, so every now and then I'd hear her working away, and zap, and I hear, damn it, zap, damn it. As you gotta fiddle with the voltage to get it right, to make sure that you don't explode your cells. You just shock them, open them up, and allow the plasmid to get in there. Okay. Once you get that plasmid inside, then the machinery inside that cell will work and make many copies. So it'll have its DNA. We've essentially added this plasmid another little piece of DNA in there as well too. So we can do that. Sometimes we might get several copies in a single cell. Sometimes we might electroporate and nothing goes in. So there are ways to test to see how many, if you have copies in there or not. You can even quantify them. Okay. So when we're testing this, particularly if we are inserting a plasmid into a bacteria, we will use antibiotics as a selection method. Okay, so our different antibiotics, penicillin, ampicillin, streptomycin, all these will kill bacteria, ideally, so we don't have MRSA. So our plasmids can carry genes that will give the host bacterium resistance. So in our plasmid, not only might we have ligated in our gene of interest when we open it and put that in there, we may have another gene in there that confers ampicillin resistance or penicillin resistance or something like that. Okay, so that means that bacteria that have taken up the plasmid will be resistant to our antibiotic, so they'll grow. The ones that maybe we shocked them and nothing came in, well, they don't have that resistance, so they'll die. All right, so our antibiotic selection will work that way. Okay, we have a particular plasmid called PBR322, which has ampicillin and tetracycline. Tetracycline? Yeah, resistance genes in there. Yeah, okay. So the neat thing is, we may have this ampicillin site has a particular cut site in the middle of it. So we try and insert our DNA in there, all right? So we actually insert it into the middle of the ampicillin resistance gene. When we do that, we take away the ampicillin resistance. So this is a different way of doing things here as well too. So here's our plasmid. We have ampicillin and tetracycline resistance. We insert the DNA. We lose the ampicillin resistance, but we retain the tetracycline resistance. We then transform our cells so that hopefully we have our stuff in here. We may get some of our regular, we have like say a single cut site. That's where this works best. We may get some of our plasmid in there that doesn't have the foreign DNA, and we may get some that does have our foreign DNA in the middle of the ampicillin resistance gene, and we may have some bacteria that don't get anything. Okay, so now what we do is we have a bunch of bacteria. Some of them have what we want, some of them have the empty plasmid, and some of them have nothing. Okay, so we can grow them up on an agar plate. Okay, so what we can do is we take our bacteria, we'll grow them up on a plate that contains tetracycline, and only the bacteria that have the plasmid in it will grow on this plate. Okay, so now I can pick colonies here and make a little map. So what I'll do is I'll pick a colony here, and I'll have two plates. I'll touch over here and I'll touch over here. So same thing on each side. Okay, so I basically kind of double up on this. All right, and what I'll see is that some of these will grow. I now have an ampicillin plate. Okay, all right, and then one that just has tetracycline on here. So so tetracycline is killing everything that doesn't have the plasmid. Ampicillin kills one that has the plasmid that has foreign DNA in it because it no longer has ampicillin resistance. If it just has the empty plasmid with no gene of interest in there, it has ampicillin resistance, so it'll grow. So what that means is that the little colonies we see here have the plasmid inside, but they don't have the gene ligated into the ampicillin resistance. But you can see I've got 1, 2, 3, 4, 5, 6 spaces here, maybe 7, 6, yeah, six spaces where there's nothing growing. And if I just put this on tetracycline by itself, I see these colonies grow here. So what this means is these 1, 2, 3, 4, 5, 6 colonies that I picked, these are bacteria that have the plasmid. They lack ampicillin resistance, so they get killed over here with the ampicillin, which means not only do they have the plasmid for the tetracycline resistance, the plasmid has the gene of interest inserted into the ampicillin resistance gene. Okay, so then I'd go back to my comparison here, and I'd pick these colonies. I'd say, hey, these are the ones I want that have my gene in there, and then we can work on expression and all that good stuff. Okay, this is, for me, this is a little bit strange because I've never done this particular one, but it relies on having like duplicate sets of plates there. Okay, and you compare the two, you kind of map out your sites. So you got to make sure that you line them up right. Wouldn't want to rotate this 90 degrees and pick the wrong one. So, but you have a way so that you can tell which way your plates are. Okay. All right. But hopefully that's not too bad. This is just all a methodology for getting a particular gene in a plasmid into a bacteria, and then figuring out if your gene of interest is in that bacteria. Okay. Okay. There are other ways we can do this as well too that are kind of simpler. They're a one-step or a one-plate process, and we'll talk about those here in a minute. All right, but first let's talk about some cloning vectors. Okay, I've mentioned the word plasmid, and all a plasmid is a bit of circular DNA that's separate from the genomic DNA. Normally, these are going to be in bacterial hosts, so we can replicate these autonomously. We basically have like little tiny genes in here. sequences of DNA with a replication site so you can make copies of it. So just copy it and go around in a circle in there, okay? So we have origins, replication, so we can use them in bacteria, we can use them in yeast. They typically, so that they need this origin for replication. They need an antibiotic resistance gene so that we can test our bacteria to make sure that we have them in there. And they need, a cleavage site or two where we can insert our genes. Okay? I love this because it says, oh, typically we can, these will allow cloning of DNA up to 15,000 base pairs. Good thing I didn't have this class before I did my graduate work, because the plasmid we had been engineered and added to and over and over and over and over again, and it was like 62,000 base pairs. Apparently it's too big to work. It worked perfectly every time. It was fine. But generally we want to try and keep this fairly small. I mean, it's hilarious because I looked at like some other people who were using different plasmids, and they'd have to set up their PCR for amplification, and it'd take them like 3 or 4 hours to run, just because I had such a huge plasmid in there. A longer sequence takes a longer time for it to amplify. My run times would be like 2 days. So I'd put in the PCR machine, come back in a couple days, let it go over the weekend or something. But yeah, you can certainly have much bigger plasmids, but optimal plasmids are going to be fairly small. Okay, this is great if we want to clone like a gene or a couple genes or something like that. But that's only 15,000 base pairs. If we look at your whole chromosome, we're talking a couple 100,000 to several million base pairs, and you can't fit that much in a plasmid. That is way too big. So there are different systems for that. We can use things like bacterial artificial chromosomes if we're gonna, our BACs, and we can use yeast artificial chromosomes or our YACs. Okay, and they basically will allow us to have larger sequences that we can clone in there. Okay. All right, so we look at our typical plasmid. Again, here we have our PBR 322. I always joke it's named for Pabst Blue Ribbons. Ooh, drink enough and come with us. Okay, terrible. You can see we actually have multiple sites in here, multiple cut sites. We have a PST site inside our ampicillin resistance. We have an ECO-R1 and a PVU2 that are outside both. We have a BAM site and a SAL site inside our tetracycline. So in this case, in the example we talked about, we left the tetracycline intact and we clearly inserted inside our ampicillin. But here we'd have an option. We could insert inside our tetracycline and leave our ampicillin resistance intact. We just wouldn't want to cut both places. Okay? And here's our origin replication. So maybe if our gene, we can cut out this way, maybe we could just insert it in an ECO-R1 site. That would be great. Okay? All right, so this is actually fairly simple, fairly small. By itself, we're talking about 4,400 base pairs. So that gives us over we could have a gene that's like 10,000 base pairs long. We can have multiple genes. We could have a whole bunch of sequence that we could fit in there. So we got lots of space basically. All right. So if we look at that, say we're going to use our ECO-R1 site, we cleave this open, we cleave our DNA, we then combine these two together, add our DNA ligase to ligate it in. So now we've added in our recombinant DNA. Once we have our ligated plasmid with its insertion in there, we can electroporate to get this into the bacteria, and then we can grow these out here. Okay. A lot of times, we may use a colorimetric approach in here. We'll attach a gene that will allow bacteria to utilize X-galactose, X-gal, and if it does that, it will turn colors. So our blue colonies will say, hey, we have our gene of interest in here. Our white colonies are, okay, we have the plasmid, but it doesn't have the inserted thing that allows it to metabolize X-galactose. Okay. So that's a nice, easy way rather than needing two separate plates to map. So it saves on resources. It's environmentally friendly, but we can just do it on one plate. You don't need to make a map, and you get like kind of one step. Hey, okay, our bacteria that are growing, the ones that have changed color are the ones that have the insert. Sometimes we'll do it the opposite way as well too. The ones that don't change color will have the insert, but it's just multiple options in there, multiple ways we can do this. Okay. If we look at our YACs, okay, they're a little bit different, but yeast are the simplest eukaryotes for DNA recombination. They have nice linear chromosomes with telomere ends, so those can be unstable in vitro. So what we do with our YAC is we make it circular to accommodate for that, so we don't have to worry about it being cleaved off each time. So So we make this circular plasmid in here. Again, just like before, we have multiple sites in here. We have our little kind of telomere sites in here as well too, our BAMs. Selectable markers, these can be resistance to maybe some type of something that will kill them, or something that will allow it to metabolize, something that will change color. So we can see our yeast change color here. But again, our yeast cloning vectors will replicate quickly because we want to have lots of them in there. And we can linearize it for transformation as well too. Okay. Basically, I want you guys to know plasmids really, really well, and then know that YACs and BACs exist that can handle larger sequences that have a little more technical detail required for them to work easily. Okay. All right, what's next here? Okay, so suppose, if we go back to my research, it's like, oh, I want to make a site-directed immunogenesis in here. I need to change something here. So a lot of times, if we make a mutation in a protein, we can figure out kind of what that protein is doing or why that sequence is important. Okay, so to do that, we will change maybe one codon, sometimes two codons, to end up getting an mRNA that's slightly different, so we can get a protein with a different amino acid in there. Okay, so we change our nucleotides in the coding DNA and express the mutated gene. So we're often going to use chemically synthesized primers that are incorporated into the DNA. If you go back to the last time we actually had class in person, or the previous recorded lecture, come to think of it, you can binge watch Dr. Ebert's Biochemistry. Okay, no, boy, that'd be something, huh? Yeah. Anyway, but our primers are just complementary sequences of DNA to introduce a mutation in there. We just have a mismatch in there. So everything matches on one side, everything matches on the other side. We just have one little section where they don't match up. So you get that kind of little hump that's drawn in there. And that mismatch can be our altered sequence. So that's our mutated primer. Okay. So our mutated plasmids are always sequenced to make sure that we have them in there, make sure we got the right sequence in there that we want. And then how do we actually make these primers and get them in there? We're going to utilize polymerase chain reaction, or PCR, which conveniently is, I believe, today's lab. Well, depending on when you view this, the lab for this week. All right. So see, if we'd actually been in class on Monday, we'd be all prepped for Friday's lab, Tuesday, whatever days. Anyway. I love the fact, I don't know if you guys ever follow along like the transcript. Whoever's doing the transcribing for me has a Herculean task, because I just have so many little fillers, and apparently my enunciation is not great, and lots of non sequiturs in there. Anyway, we're going to amplify our DNA in a test tube. Okay, so we can amplify regions of interest, usually genes within DNA, and we can amplify completely circular plasmids as well too. All right, so we mix together our target DNA, the thing we want to amplify, our various primers, which are going to be, if we have linear DNA, they're going to surround that, or they're the things that are complementary to the circular DNA. We'll need nucleotides because we're building DNA, so we need our A, C, G, and T. And we're going to need a thermostable DNA polymerase. Technically, we don't need thermostable DNA polymerase, but we heat it up to have this reaction go. When we heat it up, we'd kill our polymerase, so you'd have to re-inject polymerase after every cycle, which would get very tedious. But the discovery of thermostable DNA polymerase from organisms that live in near gas vents underwater, they may live in environments that are like 80, 90 degrees Celsius, and they have a DNA polymerase that functions there. So those allow us to heat this up and cool it down. So here's what we do. We place all this stuff together in a thermocycler, and we're going to melt our DNA. So we're basically going to raise the temperature up to 95. That's going to be a sufficiently high temperature for all of our double-stranded DNA to separate into single-stranded. We're going to let that cool, and we're going to see annealing. If we just had regular DNA in there, we just anneal back together. But now we have our primers, so we can get a primer on there, and we can start extending this way. If we have two primers on either side, we can extend the DNA in there. Okay, so we cool these anneal. Our polymerase will extend the primers in the 5' to 3' direction, and then after this is done, we do it again. We melt it again, do it again. And the fact that we will have a very, very high concentration of primers in there compared to native DNA means we are going to selectively amplify our gene of interest, okay? So we can see something like this pictorially. Here's the region that we want to be amplified, okay? We melt our DNA, we let our primers get on here, and we have our first round. So we're going to have, our long strand of parent DNA. We're going to have this, abbreviated strand that we've made in our first copy here, okay? We do this again, we get primers on here, we're going to make another long strand. So we're going to make, kind of these long strands are going to, make the same amount each time. But now, if we have a primer on here that comes to this end, we're going to selectively make a smaller region in here. And so what we do is we get, instead of we'll have an arithmetic progression of these longer copies. What we really want is the shorter stuff in here. We have an exponential growth of those. So basically, after the first, round, we don't have any of the really short stuff. So that's 0. And then we make 2 copies. Then we make 4 copies, and then 8, and then 16, and then 32. So, after like say 16 or 24 or 30 rounds of PCR, we're going to have, millions to billions of copies of our gene of interest in here. Okay? So we're just amplifying this particular region. Sometimes we make a longer copy, but we don't make very much of that. We'll only have maybe a few hundred of those after 30 rounds of PCR. Okay? So after 25 cycles, we'll typically get a 1 million fold increase in our little pieces of DNA here, our short sections. Okay. All right. So we just do this over and over again. Let the machine do it over and over again. Could you imagine having to pipette in new DNA polymerase after every cycle to a whole bunch of tubes? That would be tedious, very tedious. So hooray for technology. Okay. So once we've done this, we know our DNA will migrate towards the anode because our DNA is negatively charged. We'll have the agarose gel that will hinder the movement of this. So longer DNA migrates more slowly. Smaller DNA fragments will migrate more fastly. Ooh, man, been a long week. So we can use this electrophoresis to help us analyze our DNA to determine how pure something is, or to look at particular DNA protein interactions as well too. I think we're kind of changing gears here a little bit, aren't we? Yes. Okay. We're still in the PCR here. All right. So our PCR, again, we can use this for cloning. We make multiple copies, large numbers of copies of these proteins. DNA sequences, so we can tell if particular sequence is present in a target organism. Okay. You'll see this, for was it the FBI and the fingerprint analysis, the DNA analysis. And you got to amplify those genes so you can figure out which particular sequences are present in there. We can use this for site-directed mutagenesis as well too. We can make our sandwich primers where they match, except there's going to be like one little mismatch area in there, so we can kind of sandwich them to make small changes to that. I mentioned DNA fingerprinting in there as well too. Our short tandem repeats that are, everybody has different patterns of these short tandem repeats of DNA. If you get something like 14 to 16 different matches, that's like 1 in 8 billion. So pretty much to isolate down to one person theoretically, although DNA is not perfect. All right, it's my evil twin from the future. Okay. We can also look at RT-PCR, reverse transcriptase, so we can also convert our RNA back into DNA and amplify specific segments of that. We can also look at a quantitative PCR. We can show how relative levels of gene expression in an organism as well too. How much of this gene is being expressed versus how much of a different gene. Okay. Let's go back to our site-directed mutagenesis. Okay. So we can do this a couple different ways. Okay. We can have a site-directed mutagenesis where here's our recombinant vector. Okay. We cleave out a particular sequence in here and we insert some new DNA that has a particular mutation that we want. That's great if we have DNA that we want in here. Maybe we can get that DNA from PCR and insert it in there. That would be great. Okay. There's another way we can do this as well too. We can have this oligonucleotide directed mutagenesis. Here's our plasmid. We take some primers in here that we are going to work with, but our primers have a mismatch. That's the little X's in here. So that's where we'd have that little hump in there. But they're sufficient that on either side they'll still bind to our target DNA. So our primers bind, and we replicate those through PCR going up in there. And so we make copies of the plasmid, and we are going to preferentially make copies that have this mutation in there. So we'll still have a few that don't. So again, we'll need to test, but we can just copy the whole plasmid with these different strands in there. Okay. All right. So two different ways to achieve the same thing of getting a mutation. Okay. So you can see here we can go from AAA to CGC. Okay. All right. Quantitative PCR, we can figure out how much of something is present and how quickly it comes online as well too. So what we end up with is we have a little probe here, okay, and our probe has a fluorophore and a quenching molecule at the other end. So when our probe is not bound to something, those two ends come together in here, and our quenching molecule, turns off the fluorophore. Essentially, it's not going to emit light, so it's not going to fluoresce. Okay. On the other hand, when these are spread out and bound, you can't see them down here. Our fluorophore is over here making its fluorescence, and our quenching molecule is just floating off over here. It can't reach over there. So if we have lots of our target DNA, we're going to have a higher concentration of our probe that is bound to it. So we're going to see an increased amount of fluorescence. Okay. If we don't have much target DNA, then our probe is going to be binding to itself and quenching the fluorophore. Okay. So we can see here we have 3 different samples, and over time we can see that sample 1 is expressed at a higher rate because we see its fluorescence show up a lot faster than sample 2 or sample 3. Okay. Eventually, we get so much in there that we just kind of hit the limits of the maximum of our detection. Here's kind of our threshold of detection. Okay. Alrighty. Okay, other stuff. Let's talk briefly about DNA fingerprinting because we're still into our cloning here. So all humans have these short sequences that repeat next to each other. These are called short tandem repeats, and then it's just kind of like stuttering the same thing over and over again in there. How many repeats do you have? 7, 10, 100, whatever. These are variations in the length of fragments, and everybody has variations in here in these short tandem repeats and the numbers that they have. So if you look at several different sequences on different chromosomes, you can kind of fingerprint, and find a unique combination of lengths of these short tandem repeats that's unique to an individual. Okay, so we can figure out these fragment sizes by looking at capillary gel. It's basically the same, just a simpler way of running out multiple columns on a agarose gel. So we have lots of these locations in the body. If we match 13 of them, you get basically 1 times 10 to the 18th. So if you do everything right, and things turn out right, and you have several loci, you can basically get 1 times 10 to the 18th. If you look at the number of people in the world, there's something like 1 times 10 to the 10th. So statistically, 13 alleles, that should be a unique indicator for each person. Okay? That's again, statistics can be misleading at times. But anyway, so here we see locus 1 with allele 1. Here's our allele 2, whatever in here. We map these out. We see our different sections in here. We can see our different lengths. Okay, what do we get with this? Okay, these are ones that are typically used. If you guys like, was it NCIS or any of those crazy, terrible shows where they never balance the centrifuge, I'm no longer allowed to watch those with my spouse because she can hear my eyes rolling even if I try and be quiet. But we have these various different loci on different chromosomes in here. We check these out. Again, of course, melanin, you can tell whether it's male or female, but here's our repeat length range, and we amplify these, figure out how many alleles we see, and ideally that should uniquely identify a person. So that's why the CSI effect in criminal cases has come into effect. Everybody expects us to work out perfectly like this. It doesn't always work out perfectly. Sometimes you can only get like 6 or 7 matches, and we can say, oh well, only like 5,000 people in the world should have this matchup, so it's probably this guy. Sometimes you can have poor technicians who don't do a good job, or like that technician in, I believe it was somewhere in Virginia, maybe in Richmond, who just didn't do it and just said, "Oh yeah, they match. Oh yeah, they match." So human error or failings can contribute to false matches, and this is something, if anybody's ever interested in criminal justice, how our application goes in here. And sometimes it's terrible to be a scientist and try and deal with the law. I had a friend who was involved in an auto accident, and had degrees in physics, and so like, hey, and the other guy said it was his fault. It's like, it can't be our fault, and we did all the math and all that, and the court system did not want to hear it because we weren't experts. I'm like, please, 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 we are experts. All right, it's just anyway, I don't know. I'm kind of deviating towards politics because it's dominated by lawyers, and I'm like, maybe I better shut up and move on, right? All right, so let's move on. Maybe. There we go. Okay. Actually, that's probably a good place to stop here. Let me peek ahead and just. Okay. Yeah, this is a good stopping place for today. We'll pick up with this on Tuesday. Don't want to kill you guys with all my too much information all at once. I'll just sit here and get talking, and go. It's like, oh, I made a 3-hour presentation for my students. No, let's not do that. We'll try and keep it a little shorter than that. Besides, I got to go teach another class now too. Got to catch up. Okay, so let's stop the sharing there. Like I say, we've got an introduction to plasmids, an introduction to PCR here. We'll talk a bit more about PCR, and we'll also talk about protein expression. So far, all of our cloning, we've been looking at gene expression. So we're just looking at making copies of DNA. But in the next lecture, presumably on Tuesday, we will be talking about protein expression, where we can make multiple copies of proteins, which I find more interesting. But I am a protein biochemist, not a nucleic acid biochemist, so I have my biases there. But how do we make multiple copies of protein or make large amounts of protein? You can look at this in terms of like vaccines or different medicines or insulin. If you go back 100 years, you get insulin to treat diabetes. Maybe 100 years, 50 years, you get it from 75 maybe. You had to get it from horses or something, and it was not easy to get insulin. Now we make a recombinant insulin that we can grow up in bacteria, and you just have this factory of eggs where you got bacteria growing in there, and you can harvest that, and you can just get massive quantities of insulin to be used for managing diabetes. Anyway, I'm looking ahead a little bit too far, so that's what I have for today. Hopefully this isn't a horribly dull, tragic, terrible video, but enjoy. It is what it is. We're coming down to the end. We only got 2 more exams, right? All right. So if you have questions, as always, email me, contact me, swing in, and have a good weekend. I will see you all sometime, I hope.